Sick. Okay. So my presentation is neuroethology of turbulent chemosensory processing in benthic brachyura. And so what does this mean? So neuroethology is essentially the study of looking at neural mechanisms, specific areas of the brain, and how they typically drive behaviors, especially, I guess in my presentation, crabs. So this would look like foraging behaviors, mating behaviors, distinguishing between conspecifics, and other, I guess, brachyuran behaviors that would be necessary in survival. And so what is chemosensory? Essentially, this is utilizing chemical stimuli in making a decision, using it for some use in survival. And so essentially the chemosensory processes I will be talking about in the presentation are olfaction and distributed chemosensory. And these are analogous to our sense of smell and taste, but more specifically, it's how these brachyurans will be able to detect and utilize chemical cues to their advantage. And so these brachyurans typically live in benthic zone, which is the seafloor, the substrate, and whatnot. And they're pretty much confined to that because they don't live their lives in the water column. And so some more about that. Brachyurans are categorized as the infraorder of true crabs, and they have a couple of key characteristics that separate them from other decapods, which is their larger group that they belong to. And so, for example, they have this reduced tail upon their thorax. In males, it is kind of like a pin, very much reduced. And in females, it's where their egg sacs usually hang off of. So you can use these morphologies to, I guess, distinguish between the sexes. Alongside that, they also have 5 legs alongside their claws, but one of the legs will be greatly reduced. In the blue crab, you can see that there's this one right here, pretty reduced, mostly used for swimming. And then in this Asian short crab right here, it's almost smaller, kind of more reduced. And then in this Dungeness crab right here, also pretty reduced. Another key characteristic is their broad and flattened shells. They kind of have that characteristic of that, I guess, stereotypical crab look. And it's all seen in both of these species right here. If you could see it on the back of this blue crab, you could definitely see it as well. And so their evolved body plan is essentially becoming more common in, I guess, a lot of organisms today. And there's a big fancy word for it. I forgot what it was, but it was just really interesting to read about. Probably shouldn't have mentioned it right now. But my questions in my paper were, how does the central nervous system decode and interpret chemosensory information? How do, I guess, sensory information influence behavior, such as foraging and plume navigation? And then how do dual chemospecific pathways combine and influence behavioral responses? And so for the first chemoreception pathway in Brachyura, it would be far-field olfaction. Far-field meaning that you can detect something from far away. And so the cool thing about these guys is it's a low threshold, meaning that they can detect low concentrations of a stimuli from super far away. And then they'll still be able to orient themselves and be like, yes, food is in that direction. And it's very cool. And so they kind of do this with their antennules, and it's called their aesthetics. And these are kind of like the antennas on the top of their heads, that are kind of reduced. You can see them right here, but they kind of grab onto the chemical stimuli when it hits a certain receptor. And it's very cool. It's very cool. They can do it very rapidly, and gain sensory information within a millisecond. It's very efficient. So here's an example of it. You can see their antennules right here. It's very interesting. They kind of do a rapid downstroke and then push it back up. But then within the downstroke, water's like flushed into, I want to say the aesthetics. And because of like the quicker than velocity mechanism that it takes for them to do the quick rapid downstroke, um, it's quicker than I want to say the threshold velocity that like flow takes to transition between fluid states. So say like laminar flows, so slower flows to turbulent, which is quicker and chaotic flows. It literally switches the state and they're be able to just push that water into their receptor and send it off to the other portions of its body that categorizes this good food is or not. And so over here we have the aesthetos. they're the sensory portion I was just talking about. It's just I want to say a scanning microscope image of this upon the lateral flagellum, which is where the statistics kind of just sit. And then this PL version right here, that's labeled right here, that is the intennule itself. And so here's just the pathway of the olfactory, I guess, pathway. And so it begins at the sternus down to the olfactory receptor neurons. Those were the neurons I was talking about. And then those head down to the olfactory lobe, which is located in the deuterocerebrum, the portion of the, I want to say, upper brain in the brachyurin. And so within the olfactory lobe, it's organized into small subunits called the glomeruli. And then there are also local interneurons within this. So the local interneurons that kind of just sit rent-free in the olfactory lobe, kind of filter and kind of sift through the cues and signals and be like, yes, this is good information, and no, this is bad information. And so after it's kind of just been sitting here, it's processed to the projection neurons and other local interneurons in the protocerebrum, where the projection neurons terminate or essentially just go away. Um, and they project to the HBs, which are the hemiellipsoid bodies, which are essentially the memory complexes of brachyurins, where they see something, they remember it, and they're like, yes, I will come back here to forage. I will come back here because this is a mating ground and whatnot. And so for the second pathway of the dual chemosensory pathways, it is near field distributed chemosensory. And so essentially, this is a high threshold system, which means you need a higher and more substantial amount of concentration of stimuli to be sensed. And is a near field pathway, which means you need to be within a claw's reach to, I guess, sense the cue or the signal or the stimuli. And so this distributed chemosensory mechanism is utilized in evaluating food and the quality of it being like, is it worth it to waste my energy on it or should I pursue this? So acceptance and rejection. And it's a bimodal system. So it uses two units of input in comparison to the olfaction unimodal system. And so the units of input in this one are mechanosensory and chemosensory. And so this distributed chemosensory process is, I guess, completed all over the crab. They have mechanosensors and chemoreceptors over a lot of their body, but mostly it's upon their dactyls. So this little portion right here, their toes, the crab toes, and then they also have them on their mouth parts right here. And so we can just look at that real quick. So you can look at the mouth parts right here. There is the mandibles, obviously, and the maxillae and the maxillipeds. Very cool. That's where they just detect, I guess, a very close range food particles. And then these guys, the dactyls, they are used to sample the substrate and even taste the substrate. So they'll be tasting that sand. And so the mechanisms for this are lying in the chemoreceptor neurons, which travel to the lateral intennular neural pills. And then that is just essentially where a large portion of the central nervous system has its processing for stimuli completed alongside the median and intennular neural pills. And so that is where the chemosensory receptor neurons tend to go. However, the mechanoreceptor neurons, they end up somewhat in the same place, but not in the lateral and median intennular neural pills. They kind of end up, I want to say, processed somewhere unknown in the deuterocerebrum. That's a research gap. And some of them are projected to the protocerebrum, a separate part of the crab's brain, where mechanosensory is more known to lie and be processed, which I found pretty interesting, because there are a lot of, I guess, motor neuron interactions going on there as well. And that is where behavioral consequences are thought to arise. And so here's a picture of the brachyurin brain, an emphasis on specific parts such as the protocerebrum and the deuterocerebrum, which are just essentially regions. Here's the olfactory lobe, which is an important part, and the olfactory pathway, and of course the hemiellipsoid bodies, which are very interesting as well. And so with all this sensory information taken in from multiple different pathways, often all at once, how is this even worked with. How do you work with this? And so ionotropic receptors are the primary olfactory receptors within brachyurans, where their role is detection and discrimination. And so they're found within the olfactory sensory neurons, which are found on the aesthetics and the lateral flagellum, which I showed you earlier. And they kind of work as a ligand gated channel, with the ligands being the odorants, the stimuli, the cues, and the signals. And they bind to the receptor, and then the channel opens. And so cations, they fill the IS sensory neuron, and then transduction occurs, which means the information is off going somewhere, and then it goes to places unknown, also a research gap. And there's two types of receptors, co-receptors and tuning receptors, and they have their individual roles. And so it's very interesting. There's an issue kind of deciding with what do these ionotropic receptors do with the information. And so it's just very unknown right now. And so it's a good thing to look at the environment that the brachyurans live in. It's very important because of their sensory information and how it can be very chaotic, and how they adapt to it. So most brachyurans, they live in the benthic zone, especially on the seafloor. They can occur in deep waters, but they stay relatively coastal. So think like shallow waters to 660 feet deep. It's quite a lot of variance there, but that's just where most of them lie. And then there's a couple different things to factor into that, such as the benthic boundary layer, which are these three layers right here: the log layer, the viscous sublayer, and the diffusive sublayer. And so, of course, like I said, all of these are the benthic boundary layer, abbreviated BBL, which is crazy. And topmost layer, the log layer, most chaotic, the most stress layer, characterized by eddies and swirls, and I guess just very, very much turbulence. It's very different than the second layer, the viscous sublayer, where it's thin, there's almost no turbulence, and it's pretty laminar. So it's relatively calm. And so the third layer, it's even more laminar. There's not much going on. There's diffusive exchange occurring constantly. So there's a concentration gradient occurring. And so there's just areas of high concentration, areas of low concentration, and areas just mixing in between. It is also incredibly laminar. But it experiences turbulence from time to time because all three of these layers are probably about 10 meters, with some of these layers being 10 micrometers. So there's a lot of things to think about when these layers aren't even that large, and they can all interact at the same time. And so to categorize the study of flow, especially chemoreception, you need to quantify the fluid dynamics that benthic brachyura would encounter within the water, especially when you think about their little flicking antennules and whatnot. And so the Reynolds number is noted as RE over there. It helps predict the flow patterns in different situations, particularly laminar flows. It's much harder to predict turbulent flows. That's what I will say. And sometimes it's nearly impossible. The number itself represents the ratio of inertial forces to viscous forces, which essentially means forces that drive flow in comparison to forces that resist flow within a fluid. So whenever viscous forces dominate, a laminar flow is in place, but whenever the inertial forces dominate the viscous flows, the water is chaotic and less predictable. And so here are two examples of the different flows. First, obviously being laminar, relatively fluid and smooth in one column. And then here's the turbulent flow. And so how does climate change influence any of this? The primary issue is ocean acidification. As we know, ocean acidification decreases the pH and increases the amount of hydrogen ions being put out into the ocean, which can influence a lot of shellfish and bicarbonate making organisms. And so by decreasing the pH and putting out these hydrogen ions, it is also having the ability to alter chemical cues and signals and stimuli, which can create issues on the receptors and impaired binding at these receptors, as well as just issues changing the chemical compounds to these plumes, these signals, these cues. And so it's a little bit difficult. It's almost like playing telephone, where you're at the end of the line and you receive a message that sounds nothing like it was before. Um, and then this can also influence behavioral issues where crabs might essentially act on something when they shouldn't. There's a lot of behavioral consequences with this as well. So in short, climate change can, I guess, indirectly impact sensory and nervous system cues and how they tend to handle their issues. And another possible side effect is metabolism and how some species have increased metabolisms as climate change and ocean acidification makes it harder for a lot of animals to breathe. So the more they respirate, the more hypoxia can become an issue. And we'll get into it in the next slide, but how climate change could impact the water flow could also increase the rate of hypoxia as well. And so environmental issues can occur from climate change, as I kind of left off on the last slide. Increased turbulence in some of the laminar sublayers could potentially pose a threat, while some other researchers think all the layers will just completely become laminar. And so if all of the, I guess, sublayers hypothetically became laminar, there'd be an impact on sedimentation, sediment resuspension, and of course, not a lot of, I guess, like oxygen being put into the water that comes from the waves crashing and whatnot, which could also impact the rates of hypoxia from crabs already suffering from increased metabolisms. And so my first research question was, how does the central nervous system decode and interpret chemosensory information? And so we know it is the job of the central nervous system to differentiate and translate peripheral information. But the exact way that the central nervous system understands and decodes and then I guess like makes makes something of the information is unknown. So we don't know the code, we don't know how it does it. We just know the parts that play a role in it. And so input is often high resolution, a lot to it, many components, and it stands for what is called the olfactory decoding paradox. There's very many names for it, but essentially what it is how do thousands, hundreds of thousands of ionotropic receptors capture high resolution information and interpret them in a way that makes sense for the brachyuron to essentially respond in a way, just a singular way, in a way that makes sense for a situation. And so local interneurons in the deuterocerebrum are where cues and signals are established to influence behavior. So that gives us a clue as well into the decoding process. But the actual process, there's not much research on at all. And so my second question is, how does sensory information, sensory input influence behavior such as foraging or plume navigation? So within the olfactory pathway, it was pretty clear that it was mostly utilized for navigation as it is a far field and low threshold sense. And so there's information for it to be linked to navigation, such as the sniving mechanism, where they use their aesthetics to detect sense within milliseconds and then guide their flow, guided movement. Um, and essentially, in regards to the distributed chemosensory pathway, it's used in foraging. The dactyls of the legs are able to sample the substrate, so taste the sand and detect the water flow. And the brachyura is crossing the sediment. They aid in detecting the quality of food, as mentioned beforehand, with chemoreceptors kind of feeling around for texture, and mechanoreceptors influencing movement. So there's multiple, I guess, pathways that play a part in this. And my last question, how do dual chemospecific pathways combine and influence behavioral responses? So we know dual chemospecific pathways are integrated and play a part in behavior as the deuterocerebral interneurons interact with the aesthetics receptors and the hydrodynamic mechanoreceptors on the dactyls. However, brachyurons live in a very intermittent and turbulent environment, so they require a lot more that goes into their ability to sense the physical environment and chemical context. And so there's a lot of mystery with that. As mentioned before, the coding paradox. We're not sure how all of that information is taken in and then utilized. There's much more we need to know on top of that. The distributed chemosensory pathway, there's a lot we don't know about that. We know a lot about the olfactory pathway, but not necessarily the chemosensory distributed one. Anyway, that's it. I'm gonna try and like stop it, not as awkwardly.